The overall goal of this procedure is the in vitro synthesis of modified mRNA for induction of protein expression in cells. This is accomplished by first amplifying the plasmid insert or the coding DNA sequence and adding a poly-T tail of 120 thymidines using polymerase chain reaction. The second step is the in vitro transcription of the generated DNA to mRNA with polyadenine tails. Next, the template DNA is removed by DNA's treatment of the in vitro transcription reaction mixture. The final step is the Antarctic phosphatase treatment of the produced mRNA to remove 5' triphosphates. Ultimately, cells are transfected by the generated mRNA lipid complexes. Fluorescence microscopy and flow cytometry analyses are used to show the synthesis of enhanced GFP in cells.
The main advantage of this technique over existing methods like viral transfections is the lack of integration into the host cell genome, which prevents the risk of oncogenesis.
This method can help to produce missing or defective proteins in cells and can be used for vaccination, treatment of genetic disorders, and in the field of regenerative medicine.
Demonstrating the procedure will be Heidrun Steinle, a grad student from my laboratory.
Prior to the start of this procedure, the plasmids containing the required coding DNA sequences, or CDS, were amplified in E. coli and isolated as described in the protocol text. In this example, the CDS of interest is enhanced green fluorescent protein, or EGFP. To simultaneously amplify